Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is the place where we made learning of physiology easy, exciting, and effective. Thank you for joining me. And if you're new to this channel, you're especially welcome. And if you enjoy the content that we share, kindly click the like button, also click the subscribe button, and don't forget to turn on notifications so you don't get to miss any new content that we drop. All right, let's grow the channel together. So let's get right into today's lecture. Biliary secretion. This is part 4 of gastrointestinal secretion. We've talked about salivary secretion, gastric secretion, pancreatic, now biliary. Okay, so biliary from the word bile. That's what we are focusing on. So I know that this word is not new to you. I know in those days when we were little, when we kill a chicken, we are very careful not to puncture the gallbladder. Why? Because that's where bile is concentrated and it's very bitter. So if it stains the meat will be bitter, has this bitter taste. So that is bile. So when you have bitterness for somebody, they say you have bile. You're full of bile, bitterness. Right, so that's what it is, bile. Now, what is bile? Let's know what bile is from the physiological point of view. Now, bile is very important. Its only function, unlike the other secretions, this one doesn't have an enzymatic digestive function. That doesn't have an enzyme that digests any food substance. Okay, even though it aids digestion, but we'll see how it aids it, not enzymatically, but we'll see how it does that. So bile is produced from the liver. So this is the liver. Then it is transported to the gallbladder. This is the gallbladder. Okay, that's why in physiology and anatomy, we don't just say bladder, because there are different kinds of bladder. You have gallbladder, you have urinary bladder. Okay, so this is where the bile is mainly concentrated and acidified. Okay, it's acidified there and concentrated in the gallbladder. So it starts from the hepatocyte, bile secretion. Start from the hepatocyte. And those hepatocyte means liver cells, okay? Hepatocyte, liver cells. So they are the ones that secrete bile. But first of all, they are secreted as bile acids, okay? In the real sense of it, they are called bile acids. So they are secreted from the liver cell into what is known as bile canaliculi. Canaliculi. Okay, that's tiny ducts. They are like ducts, but they are very tiny. Bicanaliculi. So several bicanaliculi join to now form intralobular bile ducts. Okay? Then several intralobular bile ducts coalesce and join together, combine to form interlobular bile duct. Okay? So this one also combines again. So interlobular bile ducts now combine to form hepatic ducts. Now form hepatic ducts. So this is almost the final place. So these hepatic ducts, they have 2 of them. It's now just 2. So it's divided into left and right. So this is it here. So this is right hepatic duct. Then here is left hepatic duct. So right and left hepatic ducts, they join together. So at this place that is now one, is now called common hepatic duct. Okay? So after common hepatic duct, you see that it now comes out from the liver just shortly, common, and the cystic duct. This is the gallbladder. This duct here is known as cystic duct. What we're dealing now is the functional anatomy of the biliary tree. Okay, so the cystic duct joins with common hepatic duct to now form, let me draw it down here, common bile duct. Okay, so this common bile duct, if you see that something else joins it. This is pancreatic duct. Pancreatic duct. Okay, so common bile duct joins with pancreatic duct. So at this point, it's now one. So at this point, it is known as the pancreatohepatic duct. Very important that you know this thing. I can ask you as a multiple choice question. So it's the pancreatohepatic duct that opens into, this is the duodenum, the second part of the duodenum. Okay? This is the duodenum. So it's the pancreatohepatic duct that opens. So this pancreatohepatic duct is delivering both pancreatic secretion and biliary secretion together. Okay? So this point where it opens, it's a little bit widened, where it opens into the duodenum, and that is called ampulla of Vater. And no, we mentioned it in part 3. Okay? So it opens into ampulla of Vater. Sometimes this is called papilla of Vater. And there is a sphincter there that controls the movement, the secretion. And that sphincter is called sphincter of Oddi. Sphincter of Oddi. So this is what you need to know about the functional anatomy of the biliary tree. Okay? So now, we'll now ask ourselves, we've known about the functional anatomy. The next thing is to now ask yourself, what does this bile contain? Because when you know what it contains, it can help you, can guide you to know the functions. So, usually water, electrolytes, bicarbonate. Okay? And just to mention also, all these ducts you see here, they have ductal cells that secrete mainly bicarbonate and water and all of that salt. Okay? So, what happens is that bile itself, it's bile acid, and there are 2 kinds of bile acids. Let's mention them. That the liver synthesizes. One of them is called cholic acid. Okay? These are bile acids. Let's know them. The liver synthesizes this bile acid from cholesterol. Okay, number 2. This one has a funny name. Chenodeoxycholic acid. Okay, so they're called primary bile acids. Primary bile acids. Okay, so when this primary bile acids, they get to the intestine, the large intestines, bacteria, the large intestine has a lot of bacteria. It's common flora. They don't do any harm to you, okay? So those bacteria act on these primary bile acids and convert them into secondary bile acids, which is named, it converts this one, let's do it like this, converts this into deoxycholic acid. Okay? Then it converts this one into number 4, lithocolic acid. Okay? So these two are known as secondary bile acids. So these are the bile acids, 4 of them. Okay? 1, 2, 3, 4. Cholic acid, chenodeoxycholic acid, deoxycholic acid secondary, lithocolic acid secondary. So all of this is what we call bile acids. Now, bile acids don't just remain like that. They are combined with 2 special amino acids. Okay? So bile acids, they combine bile acids plus amino acids. So these amino acids, just 2 of them, glycine and taurine. Okay? Let me put it here. Glycine is an amino acid. Then taurine. So it's called conjugation. To conjugate, to combine. So the bile acid and this now form bile salt. Now call it bile salt. Okay? So this is what it gives you. So this bile salt is one of the components of biliary secretion. You know, that's what we're talking about. This bile we're talking about. The major component. This bile salt forms 50% of bile that is secreted. When you say bile, it's being secreted. 50% of it is bile salt. Okay? So, we are going to see other components and constituents of this biliary secretion after this break. All right, welcome back. So we started before we went on the break, the components or the constituent of bile. So we mentioned number 1, bile salt, which is composed of bile acid and amino acid, either glycine or taurine. Okay, so one of them is bile salt. And we say it takes up 50%. Bile is composed 50% of bile salts. So number 2, phospholipids. Bile contains phospholipids 40%. So invariably, these 2 are the main constituents. 90% of phospholipids, bile salts. And you know what is special about these 2 is that they are amphipatic. Amphipatic. That's, they have hydrophobic part and hydrophilic part. We'll tell you why it happens like that, these 2. Okay, so number 3 is cholesterol. After all, bile itself, bile acid, they are synthesized from cholesterol. This is just about 4%. Okay, then you now have bile pigments. So what are bile pigments? Bilirubin. I'm sure you've heard of bilirubin before. Bilirubin, biliverdin. Okay, So mainly bilirubin and biliverdin. These are bile pigments. Then other constituents, so many others. This is 4%. So this one is about 2%. 2%. So add everything together, 100%. But the two main organ of bile constituent, bile salt, phospholipids, and both of them are amphipathic. They have the hydrophilic part and the hydrophobic part. Now we've talked about constituents. So the important thing now we're not going to talk about, especially as it relates to these two, are the functions. The functions of what? Bile or bile acid. Now, it is well known, if there's any function you should not forget, there are two of them that you should never forget. If you forget it, it's unforgivable. Let's clean this. Number one function is that it helps in emulsification of fats. Functions of bile secretion. It helps in emulsification. This is a big word. Wow, what do we mean by this big word that we've mentioned? Emulsification of fats. Now, fats, one very funny property of fat is that they like to aggregate together into lumps, big lumps. Okay? So no matter, even if you chew fats, later they will come together and stick with themselves. So it is difficult for fats to be properly digested by those lipases. You have lingual lipase, you have gastric lipase, but the main lipase is pancreatic lipase. But even when pancreatic lipase acts on fat by itself alone, it will just touch the surface. Fats, they like gathering in lumps. So the lipase will act on just the surface. So most of it will not be digested. And if they're not digested, they cannot be absorbed. So what happens is that bile salts, they help to break this lump into small pieces. So by so doing, it increases the surface area that is available for lipase to act. It increases the surface area available for digestion. So more fat is digested. Okay? So fat, they usually come as triglycerides. Okay? So it breaks this one down into monoglyceride and fatty acid. Okay? So that's what this lipase does. But it does it very well when it is broken down. It digests more. But if it's still remaining in this lump, it's just touch only the surface. So that's what emulsification does. It's like a detergent action. We call it detergent action. Okay? So the number two important function of this bile salt is that it does not just help in the digestion of fat, to aid digestion. It's not doing the lipase is doing the digestion. It's just breaking it down so that more can be digested. It also helps in the very absorption of fat itself, of lipids. So it aids in the absorption of fats. But when we are dealing with absorption of fats, we will talk more about this. But just know that it aids in the absorption of fat through the formation of micelles, what we call micelles. Okay? Let's not go, it's a lot of explanation. So that is absorption of fat, to focus on that. So number 3 function is that bile acids or bile salts, it is noted that they also have laxative function. When you talk about a laxative, you're talking about something that helps bowel movement. It stimulates peristalsis. So that's it. Laxative movement. Then, number 4 is that bile helps in the excretion of substances that are not needed in the body. And what is that substance? Bilirubin. We mentioned it as a constituent. Bilirubin is useless to you. You don't need it in the body at all. So it helps in excretion of bilirubin, or you just say bile pigments. It helps in the excretion of bile pigments because bile pigments are not needed in the body. I think these basic functions you need to know about bile acids. So that's what they do. That's the function of bile being secreted from the liver and then the gallbladder into the duodenum. Okay? So there's something we now need to talk about. Very important. That's this diagram that is staring at us here. It's called enterohepatic circulation. Okay, so this is called enterohepatic circulation. Okay, now what is in vogue, especially in developed countries, is called recycling. Recycling of what? Of wastes, in the sense that you turn something that's supposed to have been discarded as waste and transform it into something that is still useful. Recycling. So it starts from the body. So this is a form of recycling. Look at this liver, this gallbladder, and this is duodenum. Okay? Now, what happens is that there is what we call portal circulation. I know you've heard of it before. Portal circulation. Portal circulation is very, very important circulation in the sense that this is the duodenum, okay? Duodenum. That's where this pancreatohepatic duct enters into the duodenum. Then this is cut into because it's longer. So at this point is the ileum. Note it, very important. This is the part of the small intestine where bile acid are reabsorbed into the portal circulation. Okay? So what happens is that the portal vein collects all the nutrient-rich blood from the GIT and transfers them to the liver. That's what this is represented. So when bile has been secreted here, it does its work. Those functions, the main function that we talked about, emulsification and absorption of fat. It happens between the duodenum and the jejunum. Then from the ileum, they move. When it gets to the ileum, they now get absorbed. Those bile acids, they get absorbed into the portal vein, and the portal vein goes to the liver. So those bile acids, they go back to the liver. About 90% of them. So only 10%, look at it, is lost in feces. 90% goes back to the liver. And the liver re-secretes it into this gallbladder and then the duodenum. and it goes again. 10% goes, 90% goes back. So this is called that's entero, intestine, hepatic, liver, circulation. Is the circulation. This enterohepatic circulation, you can call it the recycling. The recycling of bile salts to avoid wastage. Imagine just that this 90% is being wasted. Liver will be doing unnecessary work, synthesizing so much bile acid from cholesterol. That's waste. So it conserves all that. Just 10% is continually lost. Okay? So that is very important for you to know. So the next thing we're now going to talk about is how is biliary secretion regulated? Very, very simple. Regulation of biliary secretion. Now, it is through a nervous mechanism and hormonal mechanism, of course. And the nervous mechanism is parasympathetic nerve, which is the vagus. Remember that the vagus does 75% of the job of the parasympathetic nervous system. Okay? It's from the transverse colon that the pelvic nerve now does 25%. So it's the vagus nerve. It stimulates biliary secretion. And that's why parasympathetic nervous system, we usually call it the rest and digest function. It performs a rest. When you're at rest, then digest function. Why sympathetic? It's called what? If you can remember, flight or fright. Fright, afraid, or flight, run. Fright or fight response. This one is called rest and digest. So everything about that will aid digestion, secretion, motility, everything that favors that parasympathetic nervous system does that. So biliary secretion is part of it. Okay? Then the hormonal part of it now very interesting. There's this hormone we even mentioned in pancreatic secretion. It helps in pancreatic secretion also. No, pancreatic secretion even joins with this. So they are together. Cholecystokinin. And the funny thing about this is that this hormone derived its name from its function. And what is that function? Cholecysto. Cysto. You know, this is called cystic duct, cyst. So he's trying to tell you that the function is contraction of the gallbladder. That's where it got this name from, cholecystokinin. Okay? Also known as CCK for short. So that's what it does. It helps in the contraction of the gallbladder. Once food enters the small intestine, the duodenum, this cholecystokinin is secreted from the intestinal mucosa. Okay? And then it acts, amongst other things that it does, it acts on this gallbladder to contract the gallbladder. Gallbladder has smooth muscles. As it contracts, it ejects bile into this place. So it also favors the secretion even from the liver, especially the bile salts itself. Then there's another one that happens. Another very important hormone, secretin. This one, what it does is that it stimulates ductal secretion. And ductal secretion contains what? Mainly water electrolytes and bicarbonate. So all these ducts, we always talk about common bile ducts, cystic ducts, these ducts, and so on. So all those ducts, they have ductal cells. What they secrete, they don't secrete the normal bile. They secrete a lot of water. In fact, 50% of the volume of bile secretion is from the ductal secretion. So it adds to that bicarbonate pool that helps to neutralize gastric acid. No, but the main bicarbonate that comes from the pancreas, okay, with normal pancreatic secretion. But it also adds. So that's ductal, from the ductal cell. This secretin is what stimulates it. And something else again helps in the regulation of this. It's bile salt in itself. Can you imagine that? So it adds in a certain interesting manner. Bile salt. Bile salt helps in its own regulation, in the sense that the more bile that is reabsorbed from this place, the more it is secreted. You see that the more it is reabsorbed, the more it is secreted. It's a form of positive feedback. So the less bile that is reabsorbed, the less it's secreted. So it's trying to tell the liver that, you need, a lot of us are being reabsorbed. A lot of action is going on. So you need to secrete more bile, okay? Because it's signaling that there's a lot of biliary flow. The more it's reabsorbed, the more flow. So secrete more. So the less that it's reabsorbed is showing that there's no much. Because cholecystokinin is what stimulates this contraction. No, a lot of it comes from here. This is where it is concentrated. It contracts this and secretes it. So when food has left this duodenum, cholecystokinin will not contract this again. This reabsorption reduces and it signals the liver to calm down, to reduce. So you understand the logic behind it. Okay. So bile salt, and this action is called choleretic action. It's called a choleretic action to stimulate more bile secretion. So this one, this cholecystokinin that helps to contract, is called cholagogue. Cholagogue action. So you have the cholagogue action, contraction of the gallbladder to secrete its bile. Then choleretic is stimulation of the liver to secrete bile acid. They are related, but slightly different. Okay? So this is how it is regulated. So it's very easy stuff. Very interesting, easy stuff. Okay? So just go through it. And I've written a book on gastrointestinal physiology. Look at it here. It's called Gastrointestinal Physiology, broken down for you. And even other books in physiology, so many others, about 7 of them as at the time of this shooting, 7. So you can go check the description, but you see the links to some of this book, the soft copy. If you want, you can download it just for a token in Okada Books and read for further reading. Okay? Then also check out the description box. You see a link there of our website. Just visit it, and so that you can know many other things we do to make the learning of physiology easy, exciting, and effective. So see you in the next video.